In this session I'm going to cover epigenetics. Now epigenetics refers to changes to DNA or chromatin that alter gene expression but without altering the actual DNA sequence of the DNA. And what we're going to do is cover different forms of epigenetic changes to either DNA or chromatin that can affect gene expression. So what I'm going to cover by the end of this session is DNA methylation, and this is where cytosines within DNA become methylated, and this is specifically associated with silencing of specific genes. And this is also involved in epigenetic changes post-fertilization. So at fertilization we get whole-scale changes to what genes or what DNA is methylated, and that alters gene expression in that fertilized egg. We're also going to cover histone modifications, and chromatin is not just DNA, it is DNA wrapped around histone proteins, and those histone proteins can be modified with post-translational modifications, which are basically chemical modifications to specific amino acids, and that alters the behavior of chromatin, making it more loose and able to be transcribed, or tightly packed, making it silent. And these two modifications tend to go hand in hand. Where you've got silenced DNA due to DNA methylation, you tend to get silenced chromatin due to histone modifications. But I'm going to go through both of these. And then at the end, I'll briefly mention X chromosome inactivation, and there are some bonus slides at the end of the lecture which I might not go through in this recording, looking at imprinting. And imprinting is a very interesting phenomenon which can result in certain diseases caused by abnormal DNA methylation from either your mother or your father's DNA. It's quite complicated, and I can go through that in a tutorial if necessary. So firstly, this diagram effectively shows what a chromosome might look like from end to end. And at the end of chromosomes we have a telomere at one end and at the other end, and this region of DNA is lacking in genes, and these are protective regions of DNA that protect the important gene containing bits of the chromosome. So these are sort of protective caps to the end of chromosomes. We then have regions of DNA which are called heterochromatin, which generally are silenced and quite lacking in genes. So heterochromatin is tightly packed chromatin. Now when I talk about chromatin, we'll be looking at chromatin later on, but DNA is wrapped around histone proteins, and that forms chromatin. So when DNA is wrapped around histone core proteins very tightly, we call that heterochromatin, and it's generally silent, and these regions here are quite lacking in important genes. We've then got euchromatin, which has a much looser DNA wrapped around histones, much more easy to express genes from this type of chromatin. This chromatin can be silenced with histone modifications, as we'll come on to later, but this is where we'll find most of our important genes. Then in the center, in the middle of the chromosome, or where the two chromatids are stuck together, I should really say, is the centromere. It's another region that's very much lacking in genes. So where we will mostly be talking, when I'm talking about chromatin and genes, I'll be talking about genes within this euchromatin region, which can be silenced to more resemble heterochromatin. Now, I'm going to be talking about genes quite a lot, so it's probably a good idea to make sure we've all got a good idea of what a gene is, and this represents a eukaryotic gene rather than a bacterial gene, and many of you will have been studying bacterial genes which are very much simpler than this. So for example, the lac-operon, which I know you've all been studying, is very much more simple than this. I'm going to focus on a typical gene structure of a eukaryotic gene. So firstly, it's a good idea to have an idea of the scale of the size of a gene. And if you think about our genome of 3 billion base pairs of DNA, somewhere in the tens of thousands, maybe 20 to 30,000 different genes within there, and each individual gene can span anywhere from a few tens of thousands of base pairs of DNA up to a few million base pairs of DNA. So the largest gene is, or certainly one of the largest genes, is the dystrophin gene, and the dystrophin gene from start to finish is 3 million base pairs. That's a huge gene, and there are 90 or so exons within that gene, so that's huge. Some of the smallest eukaryotic genes just have a single exon, and they're quite unusual if they've only got a single exon, but a fairly typical gene might have, let's say an average sized gene might have 10 exons, and from start to finish, from here to here, might be spanning 50 to 100,000 base pairs of DNA. So if we start here, all of this region here doesn't contain exons, and then we've got exons separated by introns, so we've got exon, intron, exon, intron, exon. So this is a representation of a very small gene with just 3 exons. So even something with just 3 exons, if you take into account all of these regions here and down here, you could be looking at easily 20-30,000 base pairs of DNA. So this gene, the purpose of this gene is to produce an mRNA, and that mRNA can be translated by a ribosome into a protein. So if we work backwards, this is the mRNA, the messenger RNA, and the messenger RNA is going to be translated by a ribosome. Now the messenger RNA, the protein coding bits are the exons. So a ribosome will start translating from the start of this first exon. So there will be an ATG start site, and so this mRNA will be attached to a ribosome in the cytosol or attached to an endoplasmic reticulum and translated from the start of the first exon all the way through from exon 1, exon 2, exon 3. There's a 5' untranslated region and a 3' untranslated region which are involved in regulation of translation and other functions, which I'll not really go into. Now prior to this, which is the mature mRNA, this comes from a pre-mRNA, which is an RNA that contains both the exon, the 5' untranslated region, the exons and the introns, and then these become spliced out to form the mature mRNA. So this is a direct RNA copy of the gene from the transcription start site, which is where RNA polymerase binds, all the way through to the end of the gene. That will contain a 5' untranslated region, a 3' untranslated region, and exons and introns. So the introns are spliced out and those exons are shunted together to create what we call an open reading frame that goes from here to here, and that codes your protein. So in effect this is the bit that we call the gene, well we think of as the gene, because this is where RNA polymerase binds. It copies this region to produce an RNA copy. The introns get spliced out and we get our mature mRNA, but the gene is much bigger than that. The gene can have enhancer elements up here and then enhancer elements down here which enhance RNA polymerase binding. And the thing that encourages RNA polymerase to bind are transcription factors, and these bind to all of these proximal elements and upstream elements and downstream elements. So gene expression is tightly orchestrated by transcription factors. Now you will all be studying transcription factors because you all have either recently studied or currently studying the lac operon in bacteria, and the lac operon in bacteria is very much a simplified version of this. Firstly, there are no introns within the gene, but the lac operon is all about how RNA polymerase binds to the promoter region of a gene. Now all of this region here we would call the promoter region, because all of this sequence, that could be tens of thousands of base pairs, are involved in enhancing RNA polymerase to bind to the transcription start site. So we've got various regions such as the tartar box, we've got other transcription finder binding sites which all encourage general transcription factors and very specific transcription factors to switch on a gene. And when I say a gene is switched on, I mean RNA polymerase has bound here and is able to make a copy of that DNA into the pre-mRNA. So that's what I mean by a gene being switched on. Now this is a view of the promoter region of a gene and showing how these elements which are tens of thousands of base pairs away can influence whether RNA polymerase binds here. So here we can see the classic promoter of a gene. So here is RNA polymerase binding to the promoter region, and you see the tartar box there, and there's various other crucial promoter regions, but there's a whole complex of proteins here, and they are recruited into this complex, and they could be recruited in by binding to bits of DNA that are 10,000 base pairs away. So if we follow this piece of DNA, it forms big loops, and here is a regulatory DNA sequence which binds a specific protein which links into this complex, which helps RNA polymerase to bind. So a small change here can prevent this from binding, preventing this from binding, and preventing RNA polymerase from binding and being activated at the promoter. So this is why, although the gene might be, you think of it as the introns and exons, the gene regulatory region can span for tens of thousands of base pairs upstream and downstream from the transcription start site. Now the reason I'm telling you all of this is because these regions of DNA here can become modified, and they become modified by methylation of cytosines within these regions. So these regions are very rich in C's and G's. Usually it's a C followed by a G, and they often go CG, CG, CG. And we call those CPG islands. So it's a C, a phosphodiester bond, and then a G. Now the important thing about CPG islands is those are the sites that can be methylated, and if they are methylated, they often prevent these proteins from binding, which will prevent this from binding, which will prevent the whole complex from forming, which will prevent RNA polymerase from starting transcription. So a methylation event on DNA here, 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 and maybe here can prevent all of this complex from forming correctly and prevent transcription. So we call that DNA methylation. And DNA methylation is the first of the epigenetic mechanisms that we're going to cover. So here is a cytosine, here is a guanine, and you can see these two nucleotides are linked together by a phosphodiester bond. So we've got a C, we've got a phosphodiester bond, we've got a G. So we call that a CPG island, and this is unmethylated. Now using an enzyme called DNA methyltransferase, which is an enzyme which transfers methyls onto DNA, hence the name, it takes a methyl from this chemical, doesn't really matter what this is, it's just a small molecule which is highly reactive and donates methyl groups, but DNA methyltransferase takes a methyl from this and puts it onto this position here. And it's called 5-methyl cytosine at this point because it is the 5th position on this 6-atom ring. For reasons that I'll not go into, it's numbered 1, 2, 3, 4, 5, 6. So 5-methyl cytosine has got a methyl group there, whereas that is just a cytosine. So the effect of that methylation can be really important. As I mentioned on this slide, if this region here or this region here is methylated, then this transcription factor or this enhancer protein cannot bind, which means that this complex cannot bind, which means RNA polymerase cannot bind, which means the gene is silent. Now if we look at a typical promoter region and the region at the start, basically the region at the start of a gene, what's happened here is all of the CG dinucleotides, where a C is then followed by a G, have been highlighted in yellow. And around the promoter region of a gene, and particularly around the ATG start site, which is where translation would start, there are lots of CGs, and any of these can become methylated, and any of these can also be regulatory sequences that are crucial for transcription factor binding or other protein binding to enhance transcription. So this is the density of CGs in the promoter region, the region immediately upstream from the transcriptional start site, and also to some extent downstream of that as well. You get lots of CPG islands, whereas in typical non-promoter DNA, CG dinucleotides are actually quite rare and quite underrepresented. So this is your typical random DNA, this is promoter DNA, and these can become methylated to silence this gene so that we can't get transcription of this particular gene. So in this gene here, this representation of a gene, an actively expressed gene, we've got a promoter region, we've got exon 1, exon 2, exon 3, and we've got lots of Cs in here which are all unmethylated, represented by the letter C, and we get active transcription going on. Don't worry about what's happening downstream. In a silent gene, the majority of those CPG islands, Cs followed by Gs, would be methylated, and that will result in transcription inactivation and gene silencing. So what you might be wondering is, why does it have to be a CG dinucleotide? Why can't it just be cytosines that are methylated? And the reason is that when that DNA that is methylated is replicated, you want to keep that methylation status in the daughter cells upon DNA replication and subsequently cytokinesis. So here is a CPG island. It is methylated at the cytosine there, and it's followed by a G. And because it's followed by a G, on the opposite strand there is a C followed by a G. Remember, the sequences are anti-parallel, so if this is the 5' end, this is the 3' end, this is the 5' end, this is the 3' end. So a methylated C here means that there will be a methylated C opposite here. And when these two strands are separated in semi-conservative replication, so we know that in DNA replication these two strands are separated and the new copy is made, we have the original template copy here and here, which contains the methylated cytosine, and we have a new strand of DNA which is unmethylated. Now that's really important because methylation patterns need to be added on after cytokinesis, or certainly after DNA replication. So because there's a methylation there and an unmethylated cytosine there, DNA methyltransferase comes along and maintains the pattern. So that methylation there will dictate that there will be a methylation put in there. So it means that under DNA replication the methylation patterns can be maintained, whereas if this was just a CT residue, a CT dinucleotide sequence, there would be no certainty that there would be no methylation available here. So if that was CT, that would be an A, and you can't methylate an A. So you have to have CG so that both strands can be methylated. The methylation opposite the G, which follows the methylated C, if you see what I mean. So that's why it needs to be a CPG island, a dinucleotide sequence that is methylated. This means that the CPG status can be heritable. So what we're going to do now is look at DNA methylation in disease and physiological situations. And the first one we're going to look at is in cancer. So in cancer we get altered gene expression. So the cancer cells are abnormally proliferative cells and also invasive cells which can migrate and invade around the body. So these cells grow too fast and they behave very badly. Now this is some normal colon. It's a tissue section of some colon tissue, and there are some cells in here that are expressing in brown DNA methyltransferase enzyme. Now what you can see is as this normal colon progresses to a pre-malignant or a malignant and then a very, very highly malignant cancer, you can see that there's a general increase in DNA methyltransferase. And what the cancer is doing is overexpressing DNA methyltransferase so that it can alter the methylation status of key genes, and it can turn off the genes that are a liability for that cell. So what basically happens is cells which lose the ability to regulate their epigenetic status can just start silencing genes that would, if they were switched on, would kill them, but they switch them off so they survive. And we get selection of cells which have switched off a whole bunch of genes which promote the tumour growth. When they're silenced, they promote tumour growth. We call these tumour suppressor genes. So anyway, this tissue here is highly expressing DNA methyltransferase. And when we look at the CPG islands around exon 1 and upstream of that in the promoter region, we find that in DNA from the tumour, almost all of those CPG islands are methylated, meaning that this particular gene, the RAS F1A gene, which you don't need to know about, this gene is going to be silenced. If we look at DNA adjacent to the normal tissue, you see that sometimes it's silenced, sometimes it's not. That's probably just a bit of tumour DNA in that normal tissue. But if we look at DNA from normal colon tissue, we find that this gene is never normally methylated. So the RAS F1A gene, which is a tumour suppressor gene, is normally expressed in colon tissue, but in colon cancer it is silenced because almost every CPG island within that promoter region is specifically methylated. And that methylation is helped by the fact that the cancers overexpress DNA methyltransferase enzyme. Now the other major physiological situation where methylation statuses change are during fertilization. So during fertilization we have sperm DNA and we have the egg DNA, and the sperm DNA is, upon fertilization, is immediately, almost entirely demethylated. So the DNA methylation status is deliberately stripped off from almost all regions of the sperm DNA so that those genes can become re-expressed. In contrast, the female DNA from the egg is passively demethylated, which basically means as the cell divides, the DNA coming from the female is, the methylation statuses are not put back on upon DNA replication. So on the first cell division you get 50% of DNA methylation, and the next cell division 25% and 12% and so on. So that DNA is passively demethylated, as in it's not, the methylation patterns are not added back in, whereas in the sperm DNA it is actively demethylated. Now the effect of that is in the embryo, over time we see an increase in transcription because we've got all of these genes being switched back on because they are becoming demethylated. Now if we look at what goes on over, that was just showing up to the 2-cell stage. If we look what happens all the way through from fertilization through to the 8-cell stage here, through to the blastocyst, and then post-implantation, what we see is that the very, very early stages, this line here, you might just be able to make out that that's a bluish, dark blue line. This is the active demethylation of the sperm DNA, and then this is the passive demethylation of the egg DNA. And then once we get to the 8-cell stage, we've got quite substantial demethylation of both sperm and egg DNA, and then we start to build up methylation again, and methylations are put on in specific places to silence certain genes to encourage what we call differentiation. And differentiation is where different cells become different things. At the 8-cell stage, every cell is identical. At the blastocyst stage, you've got these inner cell mass cells which do a very different job to the outer cells. So the reason why these inner cell mass cells are different to these outer cells is they have different genes that have been selectively silenced by DNA methylation. Now also shown on this graph is this dotted line here, which shows the detection of various different nucleotide metabolites and degradation products, which are just markers that we can detect which show that demethylation is going on. So you don't really need to know what these are. But this is where we detect them. We detect that there are demethylation events going on because we can detect nucleotide fragments which are characteristic of demethylation occurring. Okay, so that is the end of the first part of this lecture. So this first part of this lecture is just covering the overview of the gene structure, which we've covered, and DNA methylation and how it controls gene expression. The next lecture will be, well, the next half of this lecture will be covering histone modifications and how histone modifications affect chromatin.